Jay here, and in this video, I'll explain the biology of CRISPR-Cas system. So the term CRISPR stands for clustered regularly interspaced short palindromic repeats, while the Cas stands for CRISPR associated or CRISPR associated proteins. The CRISPR-Cas system are an adaptive immune system which are found roughly in 50% of bacteria and around 90% of archaea. This system function alongside restriction modification, abortive infection, and adsorption blocks to defend the prokaryotic cells against any viral infection. Unlike any other cellular immunity or any other type of cellular immunity, which provides a generalized protection against any infection, this CRISPR immunity functions similarly to vertebrate adaptive immunity by generating records of previous infection to elicit a rapid response upon reinfection. CRISPR-Cas systems are generally defined by a genomic locus called the CRISPR array. This CRISPR array is a series of 20 to 50 direct repeats. These direct repeats are separated by spacers which are unique and also of similar length. This CRISPR array is also preceded by 80 rich leader sequences. Nearly around 20 years after CRISPR loci was identified in E. coli, spacers were found to derive from viral genomes and conjugative plasmids, which serves as records of previous infection. So sequences in foreign DNA matching spacers are called or referred to as protospacers. So, for example, this is an E. coli cell that is infected by phage 1. So, the E. coli using the CRISPR-Cas system would generate spacers that would be complementary to the genome of phage 1 and subsequently gets immunized against the corresponding phage. And in future, if the same cell gets infected with different kind of phage and using the CRISPR system, the cell would generate the spacer against genome of phage 2 and subsequently would get immunized against phage 2. So this CRISPR immunity is divided into three stages. First is spacer acquisition, second is cRNA or CRISPR RNA biogenesis, and third one is interference. So during acquisition, which is also known as adaptation or adaptation stage, the foreign DNA is identified, processed, and uh, the foreign DNA, the processed foreign DNA is integrated into the CRISPR locus as a new spacer. In cRNA biogenesis or expression stage, the CRISPR locus transcription occurs often as a single pre-cRNA and subsequently it gets processed into mature cRNA that each contain a single spacer. In the interference stage, an effector complex uses the cRNA to identify and destroy any phage or plasmid that has complementary sequences to the spacer sequence of the cRNA. These three steps, that is spacer acquisition, cRNA biogenesis, and interference, is carried out primarily by Cas proteins, which are encoded by Cas genes flanking the CRISPR array. The CRISPR-Cas system can be classified based on the presence of signature genes into six types, which is further grouped in two groups. That is group one, and group two. Among these six classes, types 1 and 3 are best studied, while class 4 to 6 have only recently been identified. Okay, so type 1 system. So the signature um, protein of type 1 system is Cas3. This Cas3 is a protein with nucleus as well as uh, helicase domain that functions in foreign DNA degradation uh, to cleave DNA that is recognized by multimeric cascade protein, uh, which stands for the protein's name is cascade, stands for CRISPR associated complex for antiviral defenses. The signature gene of type 2 system is Cas9, which is encoded by Cas9 gene. The Cas9 protein is a sole protein which is necessary for interference. Type 3 system are signified by presence of Cas10, which assembles into cascade-like interference complexes for target search and destruction. Type 4 system have a protein called CSF1, which is an uncharacterized protein proposed to form part of cascade-like complexes. Um, type 5 system also contains Cas9-like single nucleases, either CPF1, C2C1, or CTC3, depending upon the subtype. The last type, that is type 6 system, have C2C2 protein, which is a large protein with two predicted higher eukaryotes and prokaryotes nucleotide binding or happen binding RNase domain. Okay, so type okay, if I can just type 1, 3, and 4 are group 1. So this grouping is done based on the fact that this three system, that is class 1, 3 and 4, uses their multimeric effector complexes, while which means that 2, class 2, class 5, and class 6 system is grouped into a separate group. It is group 2, um, because these three um, types utilizes a single subunit effector. Okay, so if I could give you just brief about what we have gone through. So, for example, yeah, suppose this is a phage uh, containing a viral genome, and it is uh, infecting uh, E. coli, for example. All right. So when the virus attaches to the cell surface of the E. coli plasma membrane, the viral DNA is transferred into the E. coli cell. So in the first stage, if I could just zoom this out, So the first stage, that is adaptation. So this would be our 80 rich leader sequence. Then the direct repeats of 20 to 30 base pairs. And remember, the cell, the E. coli cell or any prokaryotic cell that has the CRISPR-Cas system can store number of spacers. The spacers are um, complementary sequences matching any previous infection of um, virus or plasmid. So for example, this E. coli cell was already infected with another phage, and a specific spacer complementary to that phage was already integrated into the CRISPR array. So giving it the name S1. Again, this spacer uh, would be followed by direct repeats of 20 to 30 base pairs, and that's it. So in the adaptive adaptation phase, what would happen? The CRISPR array would integrate the genome of this phage, of course, after processing. So that is S2, that is R, so that is S1, and call it. Every spacer would be followed flanked by direct repeats. So that this is R, this is S1, and again R. Okay. So this is our stage one, that is adaptation. In stage two, that is cRNA biogenesis. Uh, a pre-cRNA um, would be generated using this CRISPR array, and it would further get processed into uh, mature cRNAs, each containing the protospacer. So I'm highlighting um, this. This is our S1 protospacer. This is our S2, or the newly integrated spacer. And in the third stage, that is interference, using the crRNA, if I could just change the color, with an effector protein, and of course this would be similar crRNA that was generated using the spacer from a newly infected phage. So the defense mechanism is ready. So when in future, if the uh, phage same phage okay, the same phage will infect the bacterial cell, this CRISPR factor complex would recognize this and would break down the viral DNA and preventing the bacteria from getting infected and subsequently getting killed by uh, virus. Subsequent videos, I'll explain you about the different types of acquisition, how the CRISPR-Cas system recognizes self-genome and non-self genome, as well as mechanism through which the protospacer gets integrated, and how this CRISPR-Cas system can be utilized as uh, genome modifying tools. So thank you so much for watching this video. I hope you learned something new today. And if you have any comments, any queries, please write it down below in the comment section. And I'll see you in the next video. Thank you.